Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
My name is Dennis Gannon. I'm one of the organizers of the event, and it is my pleasure to introduce one of the co-chairs of the conference, Tony Hay. Tony is a corporate vice president for Microsoft Research Connections. Tony is a person who's been in the academic community for a very long time. He was Dean of Engineering and Chair of Computing at Southampton. I would credit him with basically inventing the term e-science because he ran the e-science program in the UK, an incredibly successful program that's still going on today. At Microsoft, he's been doing an amazing job and pulling together a lot of different university research projects and connecting them with the Microsoft Research organization. But I'm not going to say any more. Tony, it's yours.
Thanks very much, Dennis. That's okay. Okay, no, it's great to be here, and a nice day in Berkeley. When I was last here for my son's graduation in June, I think it was raining. Never rains, but it does occasionally. So it's great to be here, and I'm just here to say a few words of welcome. So first of all, I think we're all aware of the data explosion. Big data is everywhere, and almost every part of science is now engaged in data-intensive research, and increasingly we're seeing that federal agencies are taking an interest in what you do with your data, where do you save it, where do you archive it, and so on. So technology to publish and share data in the cloud is clearly going to be relevant. And then data analytics tools to analyze the data you've got are coming important, and things like machine learning as a service. And finally, in the UK, the equivalent of NSF has just asked universities to be responsible for the data that their researchers produce for 10 years. And keeping data for 10 years costs money. And so at the moment, universities in the UK are panicking about how they're going to do that. In the US, we're panicking about what a data management plan is, and so on. So there are many issues that we need to address, and the cloud, I believe, can address some of them. So I do believe that cloud data services from commercial providers like Microsoft, Amazon, Google, and so on will play a role in this and can democratize access to some of the scientific data that we have. We have to have research data services built on top of the basic cloud, so they have to be open and extensible, they have to be easily accessible, and so on. But above all, they can actually encourage scientific collaboration. So I think what I'm looking to find are examples and examples of best practice and so on. But instead of having to have massive amounts of data storage, massive amounts of support in order to maintain this, maintaining data in the cloud has a number of advantages. But again, we need to experiment to find the right sweet spot in terms of local versus cloud and sustainability. So what I'm excited about is the Cloud Futures papers here present some of the ideas of what's possible, and I'm hoping to learn a lot from what's happening today. Maybe one of these days we'll publish a sequel to the fourth paradigm when we've all figured it out. But I think it's a very exciting time, and I'm grateful to you all for coming and helping us figure out where the cloud can play a role. So this is the third workshop in a series that started in 2010, and I'm very grateful to hosting it at UC Berkeley, which is obviously one of my favorite places. So thank you to Michael Franklin, our co-chair. And we have attendees from 18 countries, apparently, and over 60 institutions. So I think that's, now, when this started, this was one of the few cloud conferences. Now there are many, so it's a testament that people are still interested to come to our conference and tell us about what they've been doing. So I'm grateful that you're here. So the program overview, in a moment I'll hand over to Dennis to introduce our first keynote, Joe Hellerstein from Google's big science effort, and I'm looking forward personally to hearing that science in the cloud at Google. And then we have Youssef Khalidi from Microsoft looking at opportunities and challenges for large-scale cloud computing. There's lots of plenary sessions, parallel sessions, and a poster session. And at the end of the day tomorrow, we have a panel chaired by Michael, who's going to talk about big data on campus, addressing challenges and opportunities across the domains. So it sounds like, to me, like a very exciting program. And what I'd like to do is just hand over to Dennis, who might want to say something about the housekeeping and introduce Juan.
Right, I'll stand down here. So first of all, I think some of you already know this is the most popular question of the morning so far. Where's the wireless? So I think that's SSID is ResComp, R-E-S-C-O-M-P, Cloud 12, password, lowercase may.
I've been overruled. See, I think you should turn off the wireless during the sessions, but
that's right.
Overruled
So
me.
it's there, but please don't use it. Okay. If you want to tweet during the meeting, there's a Twitter hashtag. The speakers, please submit your slides to us or to the front desk, because we want to make sure we have all the slides. And there's a form I think you need to sign, because some of this is being webcast as it is right now. For those of you that drove your car up here and you want to go to the dinner, we advise you not take your car to the dinner location. Leave it here. And the reason is that the parking is $4, but they only take $1 bills. So if you've got 4 $1 bills, you want to do it, that's fine. Otherwise, we're going to run the shuttle back and forth here. So if you have your car here, you go to the dinner, you can come back here, and because the shuttle will just circulate. So don't worry about that. There was some other announcements that we'll give you after the keynote, just before the break. So right now I'm going to hand it over to Juan, and Juan is going to really make the next.
My name is Juan Vargas, and with Dennis and Tony, I am one of the organizers of this conference. And it is my great pleasure to introduce Joe Hellerstein, who is going to be doing the first keynote. Joe had his PhD from the University of California in Los Angeles, and from there he went to work for 20-some years at IBM Research in Yorktown Heights, New York. Then he joined Microsoft, where he was in the developers division, working on .NET. And then from there, he went to Google, where he became the manager of Big Science. And he's going to tell us in his presentation, which is entitled Science in the Cloud, he's going to tell us about his new program, which is Computational Discovery. Joe?
So I want to share with you some work I've been doing over about the last 9 months. About 9 months ago, I was chatting with some of our senior leadership in Google, and they were asking about, well, now that we have this planetary scale distributed computing system, basically that advertising built for us, so what's next? What are the next interesting workloads? And so I started down this path trying to ask the question, well, I don't want to go to commercial companies and say, well, gee, we're sort of doing some exploration. Okay. All right, enough technology interludes. So I wanted to do something where it could be exploratory, where I could talk with people sort of freely. They had no illusions about the state of what I was doing, no promises were going to be made. And so if we wanted to look at big data and big compute, scientific codes are great for that, because most scientists today are putting their codes in open source, at least some form, and there's a lot of public data available. So in the process of doing this and talking with scientists, domain scientists, initially started out with the usual kinds of stuff, how much data, read-write rates, ratio between data access and computation, all the usual stuff we do as CS folks. But in the process of doing that and getting more detail about what the scientists were actually doing, I came to learn that there's something that's really transformational that's going on with the way, the conduct of science today and the way they're using computing. And these words almost sound sort of, platitudinous right now because they're, everybody's saying it. But it was a really interesting experience. Now, needless to say, I don't have time to share with you the entire 9 months, but I can share with you at least a couple of case studies and go through. And so actually, this talk today will be mostly chemistry and biology. There will not be that much computer science. I hope that through the process of doing this, you'll get more of a feeling for some of the issues. Over the last 9 months, I've gone from, oh, that's interesting. Let's see. Okay. Over the last 9 months, in order to be able to talk with these scientists, I found that it was really important for me to educate myself more on the sciences, too. And so that's become sort of an avid hobby for me at this point, trying to acquire this information. There's some amazing resources on the web to do this in a self-study form. So what I'm going to do, I'm going to sort of take you through a couple of case studies. I'm going to start out with a little tribute to some of the supercomputing that's going on in the cloud today, and focus on one particular piece of work we've done at Google, at least briefly, on molecular dynamics having to do with transmembrane proteins. And my goal here is to give you enough of the science background so you appreciate some of the questions that are being asked, and then see sort of this interplay that takes place between being a domain scientist and having to understand the nature of the computation that needs to be done as well. There's sort of a third dimension to this whole space, too, that I'm not going to have a chance to touch on, but that's really important. And that is, to be effective, not only do you have to understand your domain science, and you have to understand computing at scale, you also have to have sort of an understanding of statistical modeling, too. And you see that coming through time and time again. So that's another dimension of this field as well. So, okay, so I'll start out with the chemical dynamics stuff, and then I want to go into a little bit deeper into one particular area. I imagine many of you I know have been doing work in genomics, so I'm going to pick a whole different area of biology. And that's brain biology. And it's sort of interesting. We've been working with the Allen Institute for Brain Science. They're about 100 feet from my office in Seattle. And I can share with you some of the challenges that they have with some of the studies that they're doing. And then so that the computer science-y part will really be more the last part. They'll be about, after having done all this, what have I learned about the needs of scientists? And to some degree, sort of like how science really needs to change to include more concepts from computer science. Part of this is a matter of simple stuff that, is just a matter of how you do computing. But there's actually sort of a deeper understanding as well. For example, when you look at studies of the brain, one of the things that's interesting is a lot of the questions that are being asked are actually computer science questions. Biologists are asking, how is computation distributed? How do we get robustness? How do we do, how is memory stored? These are really computational questions that, you need a computer science involved. And I think that's sort of exciting, too, in terms of how we might blend disciplines. So, I don't know, I imagine many of you had seen this piece of work that was done on Amazon. This was a work that was done, really, it's a supercomputing effort done with drug discovery, and the big opportunity here was to be able to do supercomputing at a much lower cost. So these folks were able to get 50,000 cores. Largeness was done on EC2, and that was the price involved. And basically what they were doing is they were looking at molecular dynamics. I'll give you more of an example of what that means, and seeing how small molecules, some of these are actually what are called ligands, bind to the proteins. But the big thing here was to be able to do a kind of discovery at scale that was not possible before. And so this play right here is all about just providing more cycles. That's what this was about. So we've also done this at Google as well. We have a program called Exacycle. I won't go into a lot of details about the particular system that we've built. There's actually a paper coming out in Internet Computing that would describe this a bit more. But the idea behind it is that if you're able to structure your job as a large number of fairly small tasks that basically take one core, one gig for no more than an hour, then we can give you hundreds of thousands of cores. And this is not something that's available externally. It's something that right now was built internally, and so what we've done, our model is that we hire visiting faculty and they come in for a year and they have to learn about our tools and be able to do this work. But as a result of this particular piece of work, we're able to do some science that wasn't possible in many of these cases. I'll give you an example of one project. So now we're going to get into a little bit of chemistry. So before I start, I always like to ask, how many people here have a background in chemistry or biochemistry? Okay, that's actually not bad for this kind of audience. So that's good. Actually, I was recently giving this talk at University of Michigan, and I asked that question. Everybody raised their hand because I was talking to a bunch of biochemists. So that was sort of a scary thought. In any case, an undergraduate came up to me afterwards, and she told me that I did a great job of explaining these transmembrane proteins. So it was from an undergraduate, though, not from any of the PIs. So we'll see. In any case, I give you this as a way of background. Again, I'm an amateur chemist, very amateur at this point. But let me share with you, from my perspective, what the significant issue is here. So everybody's familiar with cells. All organisms are composed of cells. Well, you can argue that maybe viruses aren't, but certainly large organisms like us are. And the key thing, one of the key things about cells is how do you get molecules into the cell, nutrients, for example, and how do you get rid of molecules or produce molecules that have to go outside the cell, like into our bloodstream. And the way we do this, our cells are made up have a cell membrane around them. It's made up of what's called a lipid. It's a kind of large molecule. And the way this is done is actually very interesting. There are other kinds of molecules, proteins, which undoubtedly you've heard of. Proteins, we make many, many proteins. That's a purpose of a lot of our DNA, is to make proteins. And these proteins actually are structured in such a way that they go through the cell membrane. And the way they do this is actually very clever. Proteins consist of many amino acids. Amino acids, some of which some are positively charged, some are negatively charged. And as a result of those charge characteristics, they're able to embed themselves through this bilipid layer. Now, the next challenge is how do you select which molecules will go through the protein? And the way that works is really about the way the molecule configures itself, what's called a conformation. It has to actually orient itself into a three-dimensional shape. And that shape is structured in such a way so that the size of a molecule that can go through that protein is determined by the shape, the pores that are actually left available inside the structure, these circular structures in the protein. In addition, the protein will consist of amino acids that have the right charges and therefore will attract the right small molecules as they go through. And these proteins are very specific as to which molecules can go through. So now this flow of molecules through the protein does not happen in a passive way. Actually, our cells, and almost all big organisms do this, control this mechanism by controlling the conformation or the three-dimensional shape of the protein. And this can be done a few ways. In the brain, it's done by control of voltage. They're actually voltage-gated channels. For many cells, what happens is that in the cellular side, the intracellular space, certain chemical reactions will happen in this would be the inside of the cell. Certain chemical reactions will happen down here, which causes the orientation, the conformation of the protein to change. And so what molecules can go through will change. And that's the way the cell essentially determines what's going to come in and what's going to go out to these proteins. So a very interesting set of mechanisms. Now, what I gave you was, my simplistic understanding about how it works. The fact is that the actual mechanisms that are taking place are not well understood. A lot of these mechanisms are taking place as a result of very fine-grained time, very fine time scale actions that are happening. A lot of them are taking place in nanoseconds or even femtoseconds, which are 10 to the minus 15 seconds. And their dynamics, actually the molecules are not stationary, they're vibrating a bit. So we can use Newtonian physics and the nature of the molecules to understand those very fine-grained kinds of interactions. But the chemistry that happens, the actual transport of the molecule, what has to happen over a period of time to get molecules either in or out, that takes place on the order of milliseconds. So we've got this huge time scale difference between 10 to the minus 15th seconds and 10 to the minus 3rd seconds. We understand the rules. that will take us from this very fine-grained time constant to the coarser-grained time constant. But the time scales are vastly different. And the way we bridge that gap is computation. We just compute a lot of random trajectories over a long period of time. Now, why is this so important? I mean, it's certainly interesting from an academic point of view. But the reason why this is important is getting stuff into cells is one of the main ways we do drug treatments. We target these receptors to try and get molecules that are going to be therapeutic into cells, or things that we want to get out of cells. I mean, those are the things that we do. It turns out that this particular class of protein, these G protein coupled receptors, account for 40% of the pharmaceuticals on the market today target this class of protein. So it's a very interesting protein to study. So let me tell you a little bit about what it is that we ended up doing. So I show you the dynamics that are going on here. And the way this study was done, I mentioned before this tool, this exocycle tool that we've developed. It requires that you structure your computations into these small pieces, 1 core, 1 gig for 1 hour. But to do this kind of simulation from 10 to the minus 15th or 10 to the minus 9th seconds to 10 to the minus 3rd, you're obviously going to take much, much more time than just 1 hour, although the computations clearly fit within 1 core and 1 gig. And so the way this is done is that we're essentially we're computing what are called trajectories. We compute the molecule at one point in time. We use random numbers to sort of do perturbations, and we look at the molecule over time, look at the position of each one of its atoms in space. Once a trajectory has been computed for about an hour, then that becomes then a new computation that can be passed on to another task. And we continue on in that manner. This particular set of computations went on for several months. And the results of it were many, many terabytes of this trajectory data. And so now we got to the interesting part. We largely focus in sort of computational discovery on producing the data. Turns out we have big problems with analyzing the data, too. It's now we're in a situation where really what you want to be able to do is do some sort of interactive exploratory analysis of this data. And what we found to be extremely effective was to come up with a summary statistics for each step in the trajectory. In our case, what it was the X, Y, and Z position for each one of the atoms, because based on those positions, we could compute bonding angles. And bonding angles are going to determine the shape of the molecule, and that's going to determine what gets transported. So we wanted to have the X, Y, and Z positions for these each of the atom in the molecule over the entire period of time. And we found that basically putting that into a SQL-like database that could operate on billions of rows was a very powerful analysis tool. So what I hope I've given you so far is some sense of sort of the relationship between the problem, the chemistry problem, and the computations that we were doing. Let me also give you a feeling for, on this particular project, sort of the profile of how things went over time. So what I'm showing you here are days into the project. At this point, it's been a bit over a year, so this just shows basically the earlier parts of it. And the y-axis is the number of seconds of simulation that we've studied. Remember, the interesting chemistry is happening in milliseconds. So the more of these milliseconds we compute, the better off we are. The y-axis actually is in microseconds, so we're up to 5.5 milliseconds. So we're in the range where interesting chemistry is happening. Now, when the project first started, we had some initial data, so that's why you see it's not necessarily at 0 from some previous work, but we're well under a millisecond. And so you see this gradual ramp up. And so what we found was we were debugging a big system. Not surprisingly, we had stability problems. So our rate of progress in terms of producing useful trajectories were a little bit slow. Then we got to the point where up about until here, now we're getting to the point where we can actually look at observable data that we can compare with laboratory experiments, because now we're actually seeing the results that are the time scale where chemistry is happening. So we you can see that there's sort of this linear progress here, but it wasn't until here that we could start checking the models. We didn't have enough data to check the models until then. We just had this hypothesis about how it worked. During this period of time, we realized there were all sorts of issues with the correspondence between what we expected to happen based on laboratory data that happened in a course time scale and what we were actually observing from the computations. Once we finally got back past all of this area here, then we got into sort of a production mode where we could then continue on to the best of our knowledge about what was actually happening. So now what we're doing is we're doing this analysis I mentioned before. We have the many terabytes of data, and we're trying to understand how these models in detail work. And we're looking for internal consistencies there as well as correspondence with other literature. So what I fully expect to happen, so now we've produced over 5 milliseconds of data, is that we will find other issues out here. And that we'll be back into this space before too long. We'll realize that our models need to be refined further. So I mention this because it's always useful to get a sense of the profile that a project goes through, because then this can understand, once we sort of understand that, that becomes a kind of workflow, and then we understand the kinds of tools that we need as well. Okay, so that's the first case study. I'll come back to this a little later when I talk about what the requirements are in terms of cloud computing, being able to do science in the cloud. All right, so that's it there. All right, so let me move on to the second case study. I mentioned before some work that we've been doing with the Allen Institute for Brain Science. I don't know how many people saw this in the paper. So the Allen Institute, it's largely funded by Paul Allen's Vulcan company. And recently, they announced a new project. There's a fairly famous neurobiologist. His name is Christoph Cook. He's now the chief science officer at Allen. And Christoph, he was co-author of many papers with Francis Crick, among other things. He's very well regarded. Christoph's project is something called Mindscope, and Paul Allen recently announced that for that one project, he's investing $300 million over the next 3 years. And the assumption is that the project would actually continue on for 10. So quite an investment. So those are the folks that we're starting to work with. What I want to do in this study is talk a little bit about the kinds of problems that they're looking at. And we're a small piece of a very big effort. To do this, I'm going to have to give you a little bit of background in brain science. And the project itself has to do with sleep-wake cycles in cats. This is actually based on a paper that this was a starting point because there was some well-established science done in 2005 on looking at cats, at sleep-wake cycles. And sleep-wake cycles are interesting as a starting point to understanding what makes us conscious. And so that's an area of ongoing interest that Christoph and others has had. Okay, so how many, I'm sure everybody's heard of neurons. Neurons are the basic cell that exists inside the brain. We have, as humans, I guess the guess is something over 100 different types of neurons. They will vary in many ways. I'll discuss that in a little bit. But let me show you the basic architecture of a neuron. So we've got the cell body. It looks like a cell like any other cell. It has these additional structures going out called dendrites. And these dendrites have places where basically they can receive signals. And the structure of these dendrites, it's referred to as a forest or an arbor more commonly, is actually pretty crucial in distinguishing the types of neurons and the kinds of interconnections that they can take place. When neurons connect to one another, they connect to these dendrites in points called synapses, and an electrical current will be generated on occasion. That electrical current gets propagated across the cell body to this very critical point right here, which is not well illustrated by the original diagram, called the axon hillock. And that's the point at which, if a threshold current is exceeded, there's a current then that gets propagated all along this structure over here called the axon. And the axon is basically the output of the neuron. And the axon then has these various output pieces over here, these axon terminals, that then can connect with other neurons. So what you see here, you may notice right away, is that we have a whole bunch of potential input sources over here on these dendrites. And the outputs over here, these from the axons, can go to a whole bunch of different cells. So it's not uncommon to see interconnects on the level of 1 to 1,000. That's pretty common. So you have a massively interconnected structure. Not random, but definitely massively interconnected. Just a couple of points of reference to sort of appreciate some of the second-order subtlety, but it becomes important in terms of understanding neurons. So the way the electrical currents get propagated, I'll give that a little more detail on that in the next slide. But what's sort of interesting about this is over time, the way evolution worked is there are these insulating segments here called the myelin sheath. It's actually created by another kind of cell that wraps around the axon. And what this does is it increases the rate of propagation across the axon by a factor of 100. And you can imagine if you're working at a rate of 1/100th of your normal thinking rate, this is a big deal. So obviously that's a big advance in evolution. There are diseases that will actually cause the myelin to what's called demyelination of the axon, and it's horribly devastating, you can imagine, as a result of being able to lose that competitive advantage with transmission. That said, even though we get this big speed up by having the myelin sheaths, the propagation delays are on the order of a millisecond. Across the axon. So we're talking about a system that operates much slower than what we're used to electronically, but a system that has massive interconnects far beyond what we would normally do. Okay, so here's some frame of references in terms of the kinds of density of neurons that you see. So sort of going from quote lower level species to higher level species. So I did this in units of millions. So let's take a mouse, which is actually the focus of study at Allen is about 4 million neurons. Cat has 300 million neurons, so you can see cats should have a competitive advantage there. In general, predators do better have more neurons associated with them, and human beings were at 100 million neurons. So, and then the densities are really considerable. Our brains are a bit over a kilogram, so our densities are in the range of 1 gram of brain tissue has 71 million neurons in it. Very impressive. And that doesn't include all these interconnects as well, which are also there too. All right, so let me tell you a little bit. I'm not going to go into a lot of detail here, but I'm going to give you a little feeling about how this propagation works. The reason for all this is I want you to understand enough about the brain and the brain operation so that when we get to the point of trying to do brain science, you understand what it is that these simulations don't include, because a lot that they don't include, and you have to make a decision about what's important and what's not. So the way the signals get propagated across the neurons, it's a combination of electrical forces and osmotic pressure. And so basically, there are ions, sodium ions and calcium ions, that, you remember the G protein coupled receptors that go across the membrane and will let molecules go through? There are similar kinds of channels here that go across the membrane. These are what are called voltage gated. So remember I said before that these axons will conduct a current to another neuron, and then that neuron, if it gets enough of a voltage difference around that axon hillock, well then what will happen is these ion channels will open up. The ions will flow across, and this will create a potential difference. And what that does is then it propagates the signal across the length of the neuron. Now, this is where this axon, the myelin sheath comes in, because the way the propagation works, it turns out it jumps. You see the blue area here? That's where the myelin sheath is. It's able to jump across that fairly rapidly because of that insulation, and then it goes along over here. And the reason for the gaps between it is that actually the axons have additional structures that can go off and to provide additional connectivity. Again, some of the way that the brain has developed over time to do computation. So the key things here are that the way this propagation works, you actually have to take into account some of these ion channels. And that becomes another level of detail in a simulation of brain operation. So the last piece of this architecture at the micro level is are the synapses. So the synapse is where the axon endings over here, these terminals, connect to another neuron's dendrites. Now, there's some interesting subtleties that take place here. The way this transmission works, this is not an electrical transmission. This is a chemical transmission. There are various chemicals, neurotransmitters that are used, 5 or 6 common ones. They have different characteristics. Some of them are used to excite neurons, some are used to inhibit them. And then then one of the other interesting subtleties around this is that it's not just a matter of, so what happens is that the sending neuron on the axon side has structures inside that will release these transmitters across the cell wall. And those transmitters then get picked up by a transmembrane protein on the receptor side, and then that causes stimulation on the other end. That's where some of these voltages then can be created on the other end. So the transmitters, if you want this to work right, we're spraying chemicals into this gap, but in this axonic gap between the presynaptic and the postsynaptic complexes. And so one of the tricks to this whole process is you want that transmission to happen and then you want it to stop. Because if it just kept on pounding away, we would not get the same information being encoded. And so part of the trick to all this is that the neurotransmitters have to actually be taken up again by this same ending. And again, it goes through the same mechanism. If you have a transmembrane protein, it picks up molecules and brings them back in. It turns out that one of the effects of narcotics is that they block the uptake of neurotransmitters. And so you get this constantly stimulative effect. And you can see how then that becomes cumulative, because now then you need more neurotransmitter to have something that's a differentiated effect. So it's interesting. To some degree, if you look at this entire mechanism about how the neuron works, you and I as engineers would not have designed it like this. There's so many ways this can break down, and it is so complicated. But this is an evolved system. And so that's part of understanding the evolved system, is to understand the pieces. And one of the other challenges is then to figure out what it is that you can sort of ignore because it's part of this massively redundant system, and what is it that you really have to pay attention to actually be able to get the right biology when you're doing a model. All right, let's go at a little higher level. This is just an example of brain wiring. I'm not going to go into much detail. The only thing I want to point out is that you'll see that there actually is some sort of structure to the way these are really axons from the neurons that are being interconnected. You see if there's some sort of structure to this. There's a grouping, and it tends to be somewhat hierarchical, where you have more local connections than remote connections, and that is certainly fairly consistent across the brain. There's some macro structures that go around this. Okay, so let me take a break here and give you sort of an interesting anecdote. So needless to say, people have been studying the brain for a long time. There was a long time when there wasn't a good understanding. It's hard for us to believe this, but in ancient Egypt, there wasn't really a good understanding of what the brain did. They didn't think the brain did anything. Actually, when you were being embalmed in ancient Egypt, they actually removed your brain because they didn't think you needed it. So now obviously you're dead, so you didn't need it anyway. But it was interesting that they didn't think it was necessary. Over a period of time, and starting to where sort of modern science began to take a hold, and there was observation and reporting of information, there became to come a better understanding of the brain. And the original idea was that the brain was, we knew that thinking took place there. That was really important, but it was this amorphic mass, undifferentiated. And a major turning point, actually, was in the mid-1800s. There was a report of an incident where a railroad worker, his name was Phineas Gage, received a horrible injury. He had a metal rod that actually went through his prefrontal lobe. Now, it turns out, amazingly, the man survived. The rod was removed, but the observation was that his behavior changed radically. The initial observation, of course, the prefrontal lobe is actually where we as humans, that's one of our most evolved part of the brain. So that differentiates us a lot from primates. We don't understand all that it does, but we know that it's important. The comment from physicians at the time in the mid-1800s was, it was so fortunate that this injury occurred in an unused part of the brain. So they subsequently discovered that was not the case. This fellow, who had been a leader, very gregarious, became really a ne'er-do-well and had all sorts of psychological problems. But he did survive another 11 years. Now, I mention this only because this led to an understanding that being able to appreciate what different areas of the brain did might lead to some fundamental understanding of the brain. And that was sort of a revolution. So over time, what happened was, this is a human brain, not a cat brain. Over time, what was happening was that there's a recognition that you could actually, if scientists and physicians sort of connected examples of injuries that occurred, they could come up with sort of a map of what parts of the brain did what based on injuries they observed. Now, it's a little bit of a challenge because an injury is not necessarily localized to one particular area. So you have to draw some degree of inference. They can do more systematic studies, obviously, on animals where you can actually damage an area, cause a lesion, and be more specific. But if you want to understand the human, then you sort of take what you got. But it developed this idea of what's called Brodmann's map of the brain. This is the anterior prefrontal cortex where Phineas Gage was damaged. But there are other areas as well. For example, visual areas here. That typically there is a primary area where signals are received, and there are interpretation areas that are secondary on top of that. We have up here sensory areas and motor areas as well that have also been identified. Taking this a step further, it's also sort of interesting to see how in our evolution neurons have been allocated to function. So this is a mapping of our cortex as to the sensory areas. And you see that we have a lot of sensory area devoted to our face, our lips, also with our hands, a lot of sensory there. Not so much for our feet or much of our other parts of our body. And for motor, the same thing. We have a lot of motor areas for our hands and our tongue. So again, it is interesting to see that while there isn't a relationship between the size of an animal and the organization of the brain, that one-to-one mapping depends much more on the evolutionary value. All right, so that's my tour of brain science for you, so you have some sort of context and have some feeling for sort of the complexity of the system that now we want to model. So I'm going to take you now through this work that we've been doing as sort of a starting point with the folks at Allen. And this is modeling sleep and wakefulness cycles in cats. And I showed you a cat brain at the beginning. What we're going to do is we're going to ask the question, we have these macro level observations where we can see sleep-wake cycles, and this is actually one of the examples of the imaging. And the kinds of things that you're looking for is a cycle where there is, it's a heat map. So this is basically low voltage, no firing. These are higher voltages, and if it gets wide, it's actually being fired. And you do see this sort of oscillatory behavior in sleep-wake cycles. So that means you've got some sort of macro-level scale feedback going on. And we've got, again, these micro-level rules, right? We understand a lot about how the chemistry of axons firing. These ion channels, we understand that. We understand propagation delays across the axons. We understand a lot of detail there, the synapses. But we don't necessarily understand how the whole thing, once wired together, produces sleep-wake cycles, because that depends on a lot of the details about how much of the neurotransmitter is spread across. What are some of the places at which synapses actually occur, and therefore these voltage differentials take place? It's a lot of that detail that actually brings it all together so we get a macro-level system that actually produces something of interest to us. So that's the question. Can we actually reproduce these sleep-wake cycles? So the way it's going to do this is it's going to take this very complicated neuron, as I described it before, and reduce it to a single point. So it's just a single point, and what it's going to do is and I'll show you a little bit of the equations behind this, but it's going to take as input some of this data about current that's coming, voltage differentials from other sources, and a little bit about the ion channels, but it's going to greatly simplify what is here. The other big challenge here are the neural circuits. So cats have 10 million neurons. I'm sorry, 100 million neurons. Mice have 5. 100 million neurons, and we're going to take a small subset of that. We actually don't need the entire brain, so the cortical part is more like 10 million. We're going to take a small part of that, and we're going to then try and figure out what are representative circuits. So this is a huge challenge. First of all, trying to track down these axons and dendrites and the interconnections, it's very complicated. It's very fine-scale, micro-level kinds of observations and stainings. The second thing is there are individual differences, based on what we learned and how we developed. So ultimately, the models for interconnects are statistical models. So this is another point of essentially variation and study when you're doing this kind of biological investigation, is you've got to come up with some sort of model about what the interconnects are. And the last part is, in general, you're trying to calibrate with empirical data. And so that becomes an ongoing challenge, because at each step you want to come up with values for parameters that are representative for what the actual biology is doing. Okay, so here's sort of a summary of the kinds of parameters I mentioned before. There's a relatively simple model for propagation delay. I mentioned before there are many different types of neurons. We're not even considering the details of the neuron, and certainly not differences in types. Do a little bit with ion channels, and a little bit on some of the synaptic stuff. So the net of this is, if you want to do the simulation, you have hundreds of parameters. So this is a huge challenge if you think that you're doing science, because you've got a large number of these parameters. You're doing the best you can to calibrate to a limited amount of laboratory data, but it's all approximation. And what you're looking for is, do you match up the empirical results? Okay, so go on to a little more. I'm not going to go through a lot of detail here. There's a set of differential equations that are used to describe how the neuron behaves. This is basically how a neuron behaves. You've got this is the threshold. The threshold here is theta, and it's dynamically varying depending on this time constant. And here's how the voltages are done. Remember, the voltages are actually, it's a spatial distribution across the neuron cell. But in fact, what this is doing is saying that, well, for each one of these ion channels, so we've got a sodium channel here, we've got a potassium channel here, we'll have just simple equations to try and express those. So greatly simplified model. Okay, let's talk about, so I mentioned before, one of the other challenges is the neural circuitry. The way this was done, remember, there are different areas of the brain, and so what they did here was they developed a model about the layers of neurons that could be present, their interconnects, and had a statistical model for what those interconnects could be. And they're both excitatory neurons, and then there are also inhibitory neurons. And they indicate the inhibitions here by the black, and the red are where things are getting excited. Okay, so now you get a feeling for sort of the scope of the parameter space that we're dealing with. And so what I'm going to show you here is, if you put this all together and you do a simulation, and in this case, one of the co-authors, Sean Hill, who we've been working with quite a bit, he was the major one developing this, and you spend about 5 years calibrating this with the biology, you can actually come up with something that looks like a sleep-wake cycle. So what we're seeing here is, the potassium leak current, and as this potassium leak current starts to rise, now you see it transitioning into sleep. Things go quiet. Down here, what you're looking at is, this is essentially visual noise. It'll start coming back in a second. The visual noise will be accepted. And once that visual noise starts coming back, then you'll begin to see the rest of the circuit begin to activate again. And now you see it recovering. Okay. So how did we get here? Well, we did the best we could with the biology. We programmed it at a very fine level. We did the best we could to try and calibrate with macro-level observations. And then we did a whole bunch of parameter tweaking. And then we got a result. This is a seminal paper. And I think in many ways it's a good piece of work, but it shows you the huge gap between what scientists can actually do with these technologies and what you would consider we really have a deep insight in the discovery. And I say this, I think, I've had these conversations with Christoph Cook, who's a very well-known, I'm obviously not a biologist, but Christoph agrees. He says it's a challenging state of affairs. Okay, so like I said before, cool stuff. We got these parameters. We could actually reproduce sleep-wake cycles and satisfy a bunch of biological constraints. But there are a lot of parameters here. In statistics, there are common methods for trying to determine if you've overfitted data. I mean, like, if you have more model parameters than you have observations, then that's a real problem. Now, I don't even know how to count those here, because the observations are from empirical studies, and it's not like it's a single data point. So I don't even know how to do that computation. So I have no idea if we overfitted or not. I think the interesting thing here is that somehow they were able to search this huge parameter space and come up with something that matched. I mean, that's interesting, I mean, if they couldn't find it, that would be a challenge in and of itself. But that search is hugely time-consuming. And this is one of the areas where I think I alluded to before. I think that progress in science using computational discovery, massive computation and storage are helpful, but there are huge methodological problems. And so this idea about, how can you efficiently explore parameter space is very interesting. And then we get into some of the more details about the methodology, about there really weren't systematic ways about going about this. And the last part, and Sean actually is really good in terms of the codes he produced. Those of you who have worked with scientists and looked at science codes, especially produced by, graduate students working in domain sciences, it's a challenge to make your way through that. It's very hard. And Sean's codes were very nicely structured, but that's a rarity. Okay, so now I get into the last section here about sort of random musings. This has a little more of a computer science flavor to it. So I think, there's a lot of things that people talk about for science in the cloud. So I think this is an obvious one. Scientists are always interested in more capacity. This idea that you might rent, a huge number of cores or a huge amount of storage rather than buying it and having to maintain it. I think there's a lot of appeal to that. But I think it goes a lot beyond that. One of the huge problems today, and it's actually not limited to computational science, it's actually a big problem even with experimental science, is reproducibility. There are a lot of mentions of this, but in the computational area, there's a lot we can do to address this. So I mentioned before this issue about codes that are poorly structured. One of the implications of that is it's very hard to take someone else's code and get it to run on your system and reproduce what it is that they've done. And if we don't have reproducibility, you can ask a lot of questions about where our science is. So I think that's a huge concern. Obviously, if you can pick up someone else's code and use it, that's a huge deal. We've had some discussions from time to time with NIH, with their computational biology department, and they're saying that one of the issues they have is that there's so much resource that's wasted just basically getting to the point where you can do a study. And if instead that the underlying algorithms and data were already present in code in a common repository like in a cloud, that would be a big advantage in terms of efficiency of research dollars. And that gets the last point about that there's this opportunity to be much more efficient in terms of how we do research. Okay, so those are the appeal. There are a number of issues. I classify the issues really in two categories. First of all, there's the system stuff that we as computer scientists know. Okay, so the brain simulation is an interesting one. I work at Google. We're not known for fine-grained parallelism. We don't build supercomputers in the sense of like a Cray. So what are we doing with brain simulation? So that's actually one of the questions we're trying to explore a bit. And the initial parameters of the space were that we would just look at this in terms of how to adapt maybe some of our infrastructure to maybe do something more in the way of fine-grained parallelism. But as we got into more of the details of the science, and as you could tell, these models are very, very approximate. And for someone to say that you have to do X or Y to be faithful to the actual architecture of the brain, you really have to make an argument from the biology as to why it makes a difference. And so one of the things that we're exploring is when we make approximations, can we also do some judgment as to maybe there's a trade-off between the fidelity of what it is that's done relative to the biology, not relative to some other computation, relative to the biology, versus what is the computational advantage, like how much parallelism you can get. So I think that creates basically a whole new set of trade-offs that you might explore. One of the things that I've been very impressed with, we've worked with a number of biotech organizations in Seattle, and we as computer scientists, what we think is easy and trivial is surprisingly non-trivial to scientists, even though they have a great technical background. For example, MapReduce, or Hadoop, the same kind of programming paradigm. Going from a scientist who's working in R, just high-level statistical language, maybe does a little bit of scripting, but is not doing thinking in terms of engineering something big, to something where they have to worry about mappers and reducers. It's a non-starter. I mean, you just can't start the conversation. So one of the things we really have to pay attention to is if we're going to provide scientists with basically a low-friction situation where they can work with data, small data on their workstation, and see a relatively low-friction transition to using their same algorithms and code at scale, when now we're working with many terabytes or even petabytes, we've got some work to do because the ways we're doing it right now really are not, they're not receptive. I mentioned before in the first case study, the one on molecular dynamics, one of the huge challenges was, after we produce these many terabytes of data, how do we analyze it? And we found like a very scalable SQL was effective. Not all these analyses can be done with a sort of a declarative approach. I mean, sometimes you need an imperative language like R. So being able to do something like that at scale and have interactive performance so you can actually, explore some data is extremely helpful. This is an interesting one. So another thing that came out of the study with molecular dynamics. Remember that the progression was that, first we had to get our infrastructure to work right, and then we got to this point where essentially we're debugging the science. We had gotten a couple of milliseconds of data, enough so we could sort of figure out whether or not the science was working right. Okay, so what's going on there is that we're not doing a whole bunch of short runs. In fact, we're doing some very long runs. And what we're interested in is how these trajectories, these orientation of the molecules in 3D space, how they're evolving. And so we needed something which is sort of a combination of traditional batch, where something runs for a long period of time, with something that's interactive that can tell us, okay, here is the current state of my system. Here's what's going on, maybe even some pictures and some animation. And we've been calling this introspective batch. So it sort of combines the two of them together. And that seems to be pretty important to science because of this nature of wanting to do very long computations and wanting to see how the system itself is, if it's actually working as expected. And the last one I'll mention in terms of system challenges is I think in some ways scientists are a little bit ahead of us in the computing community in that we're really focused on our clouds. And to them, it's sort of obvious they need multiple clouds because the data are stored in multiple places. They're stored in multiple places because they're too big to move. They're stored in multiple places because of transborder data flow. They're stored in multiple places because there are proprietary concerns. There's a whole bunch of good reasons why the data will not be in a single cloud. And so this has some very interesting implications for us in terms of we ideally wanted the data in one place so that we could bring computing to the data. But if the data are inherently distributed, we've got a challenge in terms of how we build algorithms. All right, so those are the systems considerations. And I guess I can't get myself into too much trouble here because nobody here is really a domain scientist by trade. I was at a meeting last week, a couple of people, Neo and Jeffrey were both there as well, where I got myself into trouble very quickly by saying that scientists are really bad software engineers. And that their immediate response, which I had to agree with, was, well, software engineers are really bad scientists. And I agree. I didn't mean, just saying that it has some implications. But let's sort of blend this together, try and put this in a little more positive light. One of the huge challenges here is being able to develop your code in tandem with understanding with the science. This is a little different kind of thing than what we do in computing because we sort of think of more like gathering user requirements and then maybe it's a rapid prototyping cycle, but still we're not trying to painstakingly calibrate with detailed literature. The other thing which I found really intriguing is testing. So now this comes at two levels. One is the obvious level, and the obvious level is that as a software engineer, you want to make sure that your codes just do what they're supposed to do. There's another important one of this, and that is that the testing you're also interested in is if I've made a change to my model, all those things I've painstakingly calibrated to the biology or to the chemistry, the science in some way, do they still calibrate right? I mean, to us as software engineers, it seems like another kind of sort of unit test, right? I mean, it's sort of an obvious thing. It's totally foreign to the scientist. This should be part of the methodology. This is another one which I think is interesting. It's sort of like, once we get past all these problems, we can build, individual models, let's say, of chemical dynamics, then cell behavior, then maybe tissue behavior and organism behavior, and behaviors in the ecosystem. You'd like to have, an integrated stack of models. So I'm trying to figure that out. Okay, this one here is actually sort of a funny one. Okay, so there's some obvious things here about, everybody hears about scientists have all this data and it doesn't interoperate. So that's a problem. There have been a lot of attempts to try and develop common schemas. And the reason why they fail is not so much that scientists are obstructionists. They're not, I mean, maybe it's a little bit true, but it's not totally true. I think a lot of it is that the science itself is evolving, and nobody wants to be left in a straitjacket. So I don't know exactly how we accommodate that, but Bill Howe had some interesting idea about maybe a progression of schemas that might be using. So I mentioned this culture of software engineering. Okay, so this is a very interesting one about the culture one. So I had a discussion with an NIH program manager. I was actually on the side of it, but then I sort of had to insert myself, who was saying to someone else, is that I don't understand all this complaint about, software and having software be maintainable. Software languages die. All you have to do is just describe, the general principles in an article, and that's enough. And I was really frustrated. I was trying to explain to her, what this means in terms of not having reproducible results. And the one argument that really caught her attention, because she's a bench scientist by training, was saying, look, for computational discovery, software is your lab notebook. Software is where you're putting all the information about how you calibrated with empirical results. It's all in the software. It's not some general principle. It's all the detail. And that seemed to resonate with her, but I think there definitely is a cultural challenge there. Okay, that's the end of my random musings.
And I will pass microphones around. Yes, sir? Please say your name first.
I'm Neo Martinez. And hi, Joe. I was wondering, with the neural, simulations, what was the biological insight, do you think, that was achieved by that exercise?
So the main thing, there were several results that are somewhat technical, but one of them that was very interesting was the central role that the potassium leak current played. And that was, I just go back quickly, maybe I can show you just that. I didn't explain that part of the diagram so well. Okay, oops, let's go back here. Okay, so that's this chart over here, and you see how it's flat? And when it gets to the point where it shoots up, that's when we go into sleep. And there wasn't a good understanding about, at that time, about how that might be a pivotal role in terms of making this transition from sleep to wake. I'm just wondering if I can talk long enough, so it's just about to come up. Okay, now you'll see it go. So once that potassium leak current rises, now we go into sleep. And that mechanism was poorly understood. That was probably the most critical one. There were a little bit more in the way of other technical details. But I think in general what you're looking for with stuff like this is you get some insight from the computation side, and then you have to verify it in the lab in some way.
Thank you for your presentation, Ehsan Tabari. I have a question regarding your first research. You had running about a year of computation for your protein membrane analysis. How many cores were included in that, and how many proteins have you been simulating, if you know?
Okay, so
Thank you.
I gave you the time lapse of the project. Of course, there were periods of time when we weren't running anything because either our science was broken or our infrastructure was broken. But when we were running, it was the number of cores varied tremendously, but it was in the many thousands. Yeah.
Any other question? Yes.
Okay. Is there anything being done to use tomography to visualize, let's say, to analyze what is happening in real time in the brain, and then, let's say, using the cloud to try to see in real time what is happening there?
So if I understand the question is, what might be done like in real time to do analysis of the brain? There are some amazing studies that are going on for using really high-level information from the brain with, they don't even need the cloud. I mean, these are localized equipment to be able to do control. It's actually pretty impressive. And there was this month's, I think, IEEE Spectrum has something about, quote, mind reading, where actually what they've done is they've looked at fMRI studies and related that to the visual input to try and then decode essentially the transmissions that are going on inside the brain. Now, none of this actually necessarily involves the cloud, but that's very interesting from the point of view of science. In terms of like real-time data being extracted to the cloud, it's not something we're involved with. I don't know, maybe other teams are.
Okay.
Other question?
Hi, Lukas Kensel from Czech Technical University in Prague. Is there something in your compute infrastructure that you use to simulate the brain that is fundamentally different to what Google is using for other tasks, or aspects that would be different?
So with the current, and this is actually this is a direction we're going, not a direction I think we've accomplished yet. So right now what we've done is we've taken the work that this reference paper, Sean Hill and Miki Tonini have done. And we've been able to, this was code that sort of had rotted over a period of several years. We've been able to reproduce it on modern hardware. We've gotten reasonable speed up just from that. So I think the next step is to try and understand what we might do with this kind of simulation inside the cloud if we're going to scale massively. And we don't have answers there yet.
Okay. Any other question?
Okay, thank you.
Thank you.